for a lot of these things, like, it's actually really easy to make something poisonous. And as governments, as industry has grown recognition of this fact, you just have this recurring theme that all of a sudden, like, you invent a miracle something or other. Oh, plastics. Like, plastics were, thought to be the way of the future in the 1950s. They're also a type of just, molecular product. And now we find, that they choke seagulls, they choke, baby turtles. Like, there's microplastics everywhere. And, I think this is a type of generalized toxicity issue that we realize if you make large quantities of a new substance that the world broadly isn't prepared to digest, what happens is, 30 years down the line, you're like, oh crap, I killed off the trout, I killed off the eagles. So it all kind of comes down to the fact that I think, living systems are extraordinarily complicated, and making something that is tested and safe for a living thing to interact with is actually very challenging.
You're listening to Gradient Descent, a show where we learn about making machine learning models work in the real world. I'm your host, Lucas Bewald. I'm especially excited to talk to Bharath because he created the DeepChem open source project, which we've seen a lot of our customers at Weights and Biases using, and it seems to be the most popular library for people working on deep learning applied to chemistry and biology. He also made an open source dataset called moleculenet, which is a benchmark suite to facilitate the development of molecular algorithms. He got his PhD in computer science from Stanford, where he studied deep learning applied to drug discovery, and he's the lead author of TensorFlow for deep learning, from linear regression to reinforcement learning. I was really excited to talk to you. We've been seeing a lot of customers come in doing drug discovery and other medical applications, and it's something that I'm not super familiar with, but seems like incredibly meaningful. So, we've gotten a chance to talk to a whole bunch of our customers and kind of ask them what they're doing. And one thing that keeps coming up is actually the DeepChem library that I think you were the original author of. So I really wanted to start off by just asking you about that. Like, what inspired you to make it, and like what problems were you trying to solve?
Yeah, absolutely. First, thank you for kind of having me on the show. Kind of excited to chat as well. Lots of folks I know have been using weights and biases to train models and track experiments. So I think it should be a fun conversation, I hope.
Cool.
So I think a few years ago, basically during my PhD, I did a internship at Google where I used their mini computers to train some deep learning models for molecular design broadly. But I think what happened was, as with all good things, the internship came to an end. Kind of had to head back to Stanford, and then I found all of a sudden I no longer had access to all that code. I couldn't really reproduce my results. So I think the starting point was I just wanted to reproduce the results of my own paper. And I think to start, basically, it was just a few scripts in Theano and Keras at that point. And I put it up on GitHub because why not? Then a few more people started to use it, and it's just kind of grown slowly and steadily from there. I think the original kind of aim of DeepChem was really to enable kind of answering questions about so-called small molecules. So most of the drugs that we take, your Tylenols, your ibuprofens, things like that, are all small molecules. But over time, I think pharma has actually begun to shift a bit. So now there's like newer classes of medicines. There's, of course, things like vaccines. So nowadays, I think DeepChem is slowly trying to grow out to, enable open source medicine discovery across a broader swath of broader swath of modern biotech. So that's just kind of a little bit about the project. I think there is a fairly active community of users. There's a number of educational materials and tutorials built up around it. I think it's also that a lot of medicine is quite proprietary, like medicine discovery. There's biotechs, if you often like see their advertising material, be like our proprietary algorithm, our proprietary technique, which has worked fine for the industry for a long time. That's the way most medicine we know was discovered. But of course, as we know in tech, there's just been a shift in that open source is increasingly a foundational part of the way we build companies, we discover things. So I think part of the goal of DeepChem is to bring some of this open source energy to the biotech drug discovery community, and enable more people to be able to share in these tools.
And it seems like you've definitely been successful at that. I mean, I even before I knew of you, I've talking to folks at Genentech and GSK, and I would say like over half of the conversations I've had with pharma companies have mentioned DeepChem, and I thought it was pretty cool that they're all using the same platform and contributing IP. I didn't know that pharma did that at all, so that seems really wonderful.
I think it definitely is kind of a new shift in thinking, but of course, pharma's seen the fact, TensorFlow is open source, PyTorch is open source. So I think it is the beginnings of a shift. At the same time, I think like IP considerations definitely do matter a lot. So I think a lot of folks find they can't contribute at some workplaces, which is fine. I think it's kind of just a policy. But there is still a culture of caution around potentially releasing valuable IP. But I think what helps things a bit is there's this recognition that, oftentimes it's, the actual data that's the core IP. It's not necessarily the algorithm. That's just kind of calculus. And so I think there is some favorable shifts in the industry, but it's definitely something that's only beginning to happen.
So just taking a step back, because I think not everyone necessarily knows the field at all, and I actually didn't, until maybe 6 months ago when we started to see our users doing this. What's sort of the canonical problem here that pharma is trying to solve?
Hmm. Yeah, I think it's a great question. So I think, at heart, the goal really is to design medicine for diseases you care about. And the reality is, this is kind of an extraordinarily complicated process. And I'd say even now, machine learning is only useful for 10% of it. And what kind of the task here is that you say you identify a disease, then you want to find a hypothesis for what causes the disease. Maybe there's a protein that somehow has become misconfigured or mutated in the body. Maybe there's um, there can be a whole host of disease-causing factors, but you oftentimes try to take a reductionist view and narrow that down to one protein target. So you say that if I somehow could change the behavior of this protein, I could potentially cure this disease. It's a hypothesis. It might be right. It might be wrong, but it's a good starting place. Then you kind of go out and you say, all right, now I know this protein. Can I find a molecule that causes it to have some interaction? So there's a few kind of mental models for this. You can think of it as a lock and key. You can kind of think of it as basically an interacting agent that kind of comes in with the drug, that is, and shifts the behavior of the protein in a way that's favorable. So the goal computationally at a crude level is saying that design the molecule. You know, given the description of this problem, print out the ideal molecule for this. Now, the reason this gets challenging is that the ideal molecule is extremely hard. I think one of the hardest problems here is that there's this question of toxicity. So there's all sorts of I think a silly example for this is, if you want to kill cancer cells, you can pour bleach on them. It's just that you can't drink that bleach. That's going to kill you too. So, a lot of medicine is pretty indistinguishable from poison. It's really targeted poison that goes after one particular uh, part of the body. So, when you're designing medicine, you're often just struggling with this challenge of you're on this very razor thin design edge of like between poison to medicine. And you also often don't have a precise model of whether the potential drug works or not until you try it in real patients. So you try to make proxy models for this. Traditionally, you'd have something like a rat that has some variant of the disease, or sometimes it's things like cats or even dogs. And then, but when you think it's safe, you then try it out on real patients. So this is kind of the clinical trial process. There's phase one, which tests, toxicity. Is it safe for humans? There's phase two that tests efficacy. Is this actually showing effect in a group of patients that I'm trialing this on? And then phase three is basically, okay, we think there's effect. Let's make sure on a big trial with lots of people. And occasionally there's things like phase four, which is after the drug is being used by real people. Let's do more studies to understand the real effects it's having on patients so we can give better guidance to doctors. So I think the heart of the challenge in applying machine learning here is that we are dealing with a lot of unknowns. We don't know uh, precisely why things become poisonous. We know some of the reasons, but oftentimes you'll get just these strange factors that crop up. We don't know if a potential medicine actually treats uh, the disease in question until we try it. So
Well, I think
the
wait.
goal
So
is,
just to slow down for a second, I think it's not even obvious to me necessarily what the machine learning problem is within that. I mean,
Yep.
what's the input data and what's the what are we trying to predict?
That's definitely another great question. And usually the challenge here is that you start with a very narrow sliver of these problems. So there are, say, limited models for toxicity that, given some amount of data, maybe you have a database of compounds and you're like, this molecule, induced negative effect, something. You can train a machine learning model that, given the structure of a new molecule, will predict an output, which is, the toxicity label. The challenge, of course, is generalization. Uh, it works on your training set, but if I give you a new molecule, does it actually work? That's often the question. It's just very hard to gauge that.
And
Um,
then how is it possible? Sorry, dumb questions. I'll just
no, no,
ask
no.
the
It's
questions I
great.
honestly have. So, like, how would you possibly have enough training data? Like, you're not gonna keep
Yeah.
poisoning cats, right? To keep finding more and more poisonous molecules. Like, how does that work?
I think this is another great question, and the real answer is we don't have enough training data, which is why I think molecular machine learning is a bit of an art right now. Unlike images and speech, where there's this dramatically larger training sets, the data sets are fundamentally limited. There's a few approaches people take to deal with this. I think kind of one common theme is let's use more of the fact that we know a lot about physics and chemistry. Toxicity, I think, is a very hard problem because it's biology. It's
Mhm.
uh, uh, it's kind of harder. But in many cases, you'd say that, well, okay, I know something about the molecule. I know something about its invariances. I can encode that into the kind of the convolutional network. So now you have kind of increasingly sophisticated, graph convolutional networks that encode more factors of known molecular structure. Um, it's it's definitely not a solved field. I think this entire kind of part of machine learning is far from what I'd call the ImageNet moment. So that there's that point at which something just crosses over and breaks out. And I think right now it's useful, but it isn't that magic bullet in
Well, I
this
want to
corner.
actually really would like to go back to that, but I want to make sure I understand the core problem here. So it sounds like you have like a molecule and you want to predict some kind of property.
And I think that is definitely the most common one. There's a number of variants to this. Like you might have a protein and you want to find a molecule that interacts with it.
Mm,
One way you can do this is the property is does it interact with the protein?
mm,
There's also generative models where you say that, okay, given a database of known drugs, use some LSTM or something to just print out a new potential drug. This tends to get a little hairy. It's kind of hot research, but it's not safe to really use in production, I think. There's some raging academic debates about that right now.
Okay, but sorry, can I
Yeah.
ask some more dumb questions before
No, no,
you?
please, please.
So, how do you even represent a molecule like text? It seems kind of obvious to me, but like, how do you? I mean, it seems like the molecules are variable length and they have some structure.
No,
Is it a graph? Is that how do you?
it's actually a great question. So thankfully that there's kind of the field of chemoinformatics where a number of years ago they defined a language called SMILES, SMILES. So SMILES strings are basically a language that allow you to write down molecules. It's most often used for small molecules, but you can write pretty big arbitrary molecules as well. So there's a number of architectures. Many architectures take the SMILES and do convert it into a graph.
Hmm,
And the idea is that the atoms in the molecule turn into nodes in the graph, and bonds usually turn to edges. Although sometimes you do something like a distance cutoff because there's these non-covalent interactions. So you might say all atoms that are yay close to each other are now bonds in my or have edges in my graph.
hmm. And does that completely represent a molecule?
So honestly, not at all. So like the real molecules are these very complex, quantum beasts that have orbitals and like extremely complicated wave functions. In fact, I'd say that when you get past like really teensy molecules like helium, or like there's probably a few like slightly more complicated ones. You actually don't know the quantum structure of these things until I think the quantum computers like arrive and like we can run these simulations. We actually do not really have the ability to grasp the quote unquote true structure of a molecule in most cases. So it's an approximation.
Hmm.
It's mostly useful for many purposes though. But yeah, molecules are more complicated than we understand in many cases.
But when you talk about like an LSTM generating a molecule, it's generating sort of literally generating like a string that gets interpreted as a molecule.
Exactly. So the smiles language I mentioned. So precisely what you do is that you just treat it like a sentence generation task, but you're generating in the smiles language.
Mm.
Uh, and oftentimes the challenge there is that if you do this naively, you'll generate grammatical errors. So it's not an actual molecule. But there's been a lot of research. There's some groups at MIT in particular and U Toronto that have worked out ways to constrain the generative models so that it's more likely to generate real molecules.
So I guess this sounds, as like an ML person, this sounds like incredibly appealing, right? Like a kind of a well-formed, tricky ML problem that has the potential of saving lives. And I guess I wonder, like how much of this is real and how much of it is speculative. Like, can you point to an example of like a drug that was created through this process or helped by this process?
Absolutely not, unfortunately. So this is kind of where it gets really fuzzy because it's on average like, I think COVID might actually speed up discovery in some cases, but most of the time it's like 15 years from the first discovery, starting a project to like the actual getting to patients. So there have been simpler computational techniques in use for decades now. So there's some degree of evidence that they help, but I don't think there's been a smoking gun. There isn't like one molecule that you can really point to and say, that, an AI made that. And I think it's more like, the process of using this program helped, in some fuzzy, hard to quantify fashion, the design of this uh, compound.
But it seems like the programs are kind of suggesting, or at least the framing that I hear from a lot of our customers is the programs are like suggesting compounds to try, which makes a ton of sense, right? Because you have to try something. So I assume that
Mhm.
people have some non-random approach for this, right? And so is that actually better? That I guess is the do you, it seems like there must be evidence now if these deep learning techniques work better for this kind of suggestion than other techniques. Like that seems like pretty quantifiable, or am I missing something?
So I think part of the challenge here is that it's hard. There's like many steps in the process. So there was a paper from Google recently where they showed that on one particular task that when they ran the experiment, the assay naively, it was like a few percent hit rate. That is like things that actually like looked like they might work in that stage. And when they bootstrapped it by training a machine learning model, then made predictions, it was something like 30%. And
Mhm.
that sounds like a giant boost, but I think that's like one step out of like 20 in the process.
I
So
see.
so you take the thing that comes from that, then you go to the next stage where you're like, well, okay, this molecule is good, but it turns out that it gets caught up by the liver. We need to like change it somehow so that it avoids that. And right now, the best way to do that is still to hire a seasoned team of medicinal chemists who can guide you through that process. In the later stages, it gets particularly gnarly because you have very small amounts of data. So, like
Mm.
the Google paper, it was at an early stage where they could generate programmatic large data sets, like 50 million data points or something. But in the later stages, you might have like 100. And then all of a sudden, you're in that fuzzy no man's world in which machine learning is kind of witchcraft. That's, I think, part of the reason because, like, maybe you started out with something that was you know, AI generated. But then 10 medicinal chemists came along, tweaked it here, tweaked it there. Then what do you have at the end? And honestly, we don't know. Like, I think 10 years from now, maybe there will be a molecule we can point to. But for now, I think it's still fuzzy.
It's kind of interesting that you said. I mean, I totally resonate with the ImageNet moment because I definitely remember the ImageNet moment for vision because I was running a company that was selling training data and suddenly everyone flipped from wanting text training data
Hmm.
to images because suddenly all the image applications were working. But I guess what was kind of interesting was that I actually feel like the ImageNet moment came a few years after ImageNet. Like
Mm.
like not only did it have to, we saw vision starting to work, but it took people a while to realize it, and then companies started to staff up, and now, I can go on Pinterest and click on stuff and buy them right away, or I can
Yep.
like find all my baby photos in my iPhone. But like, it seems like this one, the medical companies have kind of staffed up maybe before it's clear that it's working, right? Because it does seem like deep learning is now important to basically every pharma company. I mean, it seems like this could be set up for a real serious disappointment also.
I think no, I think that's very kind of insightful as an observation, and I think you're totally right. I I think if you talk to a pharma veteran, they'll talk. There's like this old Fortune magazine from 1980 where I don't know they had some picture of molecules on a computer, and they said it's going to be like medicine on the computer. It's going to change everything, and of course nothing changed. Um, and I think even for the Human Genome Project, there's a lot of hype. People thought having access to the genome would change everything, but I think the recurring theme of biology is that it's billions of years of evolution always have more tricks behind them. So I think you're right. I think deep learning is a useful but not magical tool in this space right now. And I think that, in some cases that disappointment has already hit people. I think in other cases, though, like my hope is that people stick with it because I think these techniques do have a lot to offer. But yeah, I don't think it's going to magically cure cancer. I think it'll be one useful tool in the scientist toolkit to discover medicine.
But what do you think? What do you think caused people to feel this optimism? Because machine learning techniques have been around for quite a long time, and presumably people were trying these on the same data sets. Like, is there something special about deep learning that it sort of feels more promising in some way?
It's a great question. I think we all saw this like amazing wave of just deep learning hype. Because I think that ImageNet moment it spread out into all these other fields, and I think people started hoping. Um, and I think there are some genuinely new advances that deep learning on molecules has engendered. For example, I think the more predictive models, when you have enough data, they actually start working considerably better. This Google paper I mentioned a while back, it actually gets like a considerable boost over a simpler random forest or something because it has enough data. The generative models, they can sometimes do clever things. So I think there is some substance. It's not all vapor, uh, but there isn't that. I think there is the hope that it might lead to a breakthrough. And for just speaking for me personally, when I started working in this field, I didn't really understand any biology or chemistry. I think last ninth grade bio class was my last formal training in the subject. Um,
You
and
and me
when,
both.
yeah, I had a good ninth grade bio teacher, but yeah, I think when you come in, you're like, well, tech can solve many hard problems. Like, why can't it solve this? Why not? And I think the answer is evolution has had billions of years, and that just builds up irreducible complexities sometimes. So, I think I think I'm still hopeful. I think there is real potential and value, but I think also like once you've gone spent some time in it, I think you get some humility of this. The scope of the problem is much grander than you. At least I first realized when I was coming into the space. Um, but yeah, I think it's just the hype train got ahead of the actual technology, and then it's like Gartner hype cycle. I think now we'll end that trough of disappointment, and then that slope of enlightenment coming up a few years from now.
Interesting. People seem fairly optimistic for a trough of disappointment. So it's an
Maybe
interesting
still,
perspective. Yeah, maybe we're still coming down. I hope not. Do you
yeah.
I mean, one problem that I've always found in health applications is missing data. Like, are there data sets like ImageNet for these kinds of applications?
So honest answer, not really. So I kind of started a project called moleculenet a number of years back in grad school, along with kind of one of my co-authors. And our intent was to gather as many data sets as we could to try to make something like ImageNet. And I think the honest answer is we helped a little bit. I think there's a useful collection of data and benchmarks we put together, but the challenge is that molecules are not. So I think in computer vision, I think object detection, object localization, uh, they don't cover all vision tasks. I think there's some hard frontier problems still, but you get like a pretty big chunk of them. Uh, in molecules, it's more like there's just an entire range of things people want to do with them, and you have a little bit of data for each task, but there's like And the tasks are often not related. So if you take like a quantum mechanical data set, you'll find that very different, featurizations and algorithms actually work better than if you take a biophysical task or a biological task. Um, so I think there is a reasonable amount of data in aggregate, but it's for different applications, and you can't easily blend it into one ImageNet style mono data set yet.
Interesting. It kind of reminds me of like natural language processing with all of its different applications, I guess.
I think I think there is a dream that maybe we can figure out some type of universal pre-training that akin to the GPT-2 models or the like
Mhm.
actually does get you to that universal molecular model. I I think as of now we haven't achieved it, but maybe it's not crazy to think that we can. Like we do know that Schrodinger's equation at some deep level is pretty close to a, leaving aside relativity, it's the best known model of these molecules we have. So maybe the quantum computers will eventually help solve this, but it's a ways off for now.
Interesting. And the experiments presumably are kind of expensive to run now.
Yeah, I think there's the rise of I think mail order services, things like Enamine or Mushi. I think where you can pick out a molecule out of a catalog, then they'll make it for you and they'll ship it to you. So it's a little easier than it used to be. You don't actually need to be uh, a bench chemist. At the same time, you still need to run an experiment. So oftentimes people will say use Enamine to buy it, then they'll use a second contract research organization to run the experiment, and they'll just keep track of quality control. So it is possible to do it, you know, not quite in your basement, I think, but maybe in a well-stocked garage where you can carefully, coordinate many email threads or something like that. Um, but yeah, it's expensive. It'll put you somewhere between a few hundred to a few $1,000s per compound, depending. Uh,
So how do these startups that I mean, because we have a whole bunch of customers that are startups, doing this type of thing. How do they how do they hope to kind of compete with bigger companies when they don't have access to these data sets?
that is a great question. In many ways, maybe I'm not the right person to ask because I didn't found one of these startups.
Sure,
Uh,
fair.
I So I think there is some advantage to coming at it with some new eyes. I think when you're a very big company and are trying to introduce just a shift in thinking, there's
Mhm.
of course a lot of cultural inertia, just traditional startup versus big co dynamics. I think there is some potential to pick up kind of interesting, uh, potentially low hanging fruit that just people haven't looked at. Um, I think there is this also some eventually, I think, potential for mergers and acquisitions. I think building a talented machine learning team can be difficult. And I think if you have a company that has succeeded and has shown some promise, maybe it's a good acquisition target. So I think there are fruitful paths forward for many of these companies. I think some of them are actually aiming really high. They want to be the next Genentech. And I think it is possible, but I think that might end up coming down more to your biologists than it does to your machine learning people. And perhaps I'm a bit of a pessimist on that front. I think core biology, the really foundational stuff, is still beyond our current machine learning and AI techniques. I think it's beginning to change. I think as you get more genomics data, more kind of biological material that you can feed into machine learning models, there's a lot of companies at that frontier. But for now, I think it really is that if you have a crack team of scientists, that might take you further than a crack team of machine learning engineers. Ideally, you have both, and then you have the best of all worlds.
Although it just seems like the data collection process is so hard, it seems like you might need to innovate there too. I mean, I'm
Yeah.
coming from my own background of data labeling, but it seems so daunting, the idea that you have to like order molecules somehow and like run a
Yeah.
wet lab or something. I mean, just um, I guess okay. I have a whole bunch of different questions. I hope you don't mind. One thought I have, I guess, this is probably like the dumb things that people think of when they first hear about this stuff. But it seems like if you could model things about molecules, that's like so powerful. Like that's like the stuff everything's made out of. Like there must be applications besides biology that might be simpler. Is that true?
I think absolutely. Now, unfortunately, I think the challenging part is some of the most interesting applications are in places like batteries, and of course,
Ah.
batteries. Uh, So I think there are kind of other fields, like for example, it turns out well, the crop protection industry. So if you make pesticides, herbicides, fungicides, pretty similar techniques.
Really?
Um, mhm. So it
Yeah,
actually
I guess it's just
there's
properties of molecules.
and in fact, and this is kind of coming back to that, thin line between poison and medicine. A
Uh-huh.
pesticide, if you actually take a look at some pesticides and you look at them, they kind of look like the same small molecules you have in medicine, which might explain a few things about the world. Just saying there. Um, I think there's also other applications like I think in industrial applications broadly in petrochemicals, even I think there's a bit. It's so there's absolutely kind of other cases, but I think we in the software industry are sometimes used to working in our world of bits, and uh, whereas I think like when you get into these industries, you're like at the end you have to make something, and I think there is that slowdown. I think maybe batteries is actually the hardest. Pharma is a little behind that. I think some of these agricultural applications are a little easier to get to market, but still quite daunting. um, and I think in general it just kind of comes down to like for a lot of these things, like it's actually really easy to make something poisonous, and as governments, as industry has grown recognition of this fact, you just have this recurring theme that all of a sudden, like, you invent a miracle something or other. Oh, plastics. Like, plastics were, thought to be the way of the future in the 1950s. They're also a type of just, molecular product. And now we find, that they choke seagulls, they choke, baby turtles. Like, there's microplastics everywhere. And, I think this is a type of generalized toxicity issue that we realize if you make large quantities of a new substance that the world broadly isn't prepared to digest. Uh, what happens is, 30 years down the line, you're like, oh crap, I killed off the trout, I killed off
Mhm.
the eagles. It all kind of comes down to the fact that I think, living systems are extraordinarily complicated, and making something that is tested and safe for a living thing to interact with is actually very challenging.
Mm. What about other medical applications? Like I think you wrote a book on this, right? So like what are the other categories of things? And I guess I'd be curious your take on like how promising they are since I it sounds like it's hard to separate the hype, but you've probably thought deeply about this.
I definitely think there's a whole host of really promising applications. I think to name two, I think microscopy is going to be completely changed by ConNets. This is one of those magical places where hey, ImageNet works, as in you can actually take an ImageNet model and stick it
Hmm.
on top of a microscope and start doing pretty sensible things pretty quickly.
Hmm.
Um, so
What's
in
an example
general,
of a thing that you might do with microscopy?
so it's actually so one of the kind of interesting things about this field is that you can pick up a lot more out of a microscope than you kind of thought. So there's some really interesting papers that show that oftentimes, like, so there's some say readouts of a cell that where traditionally you have to kind of destroy the cell, blow it up in order to get at it. But people have started to show that you can instead, get a data set where you take the original cell, then you like blow it up, get the readout, but then you can train a machine learning model to start to impute that from the raw cell. So you can potentially get non-destructive readouts that enable new things. And this is kind of more basic science. Like it's not clear what the downstream effect is. There's kind of a number of companies. I think Recursion Therapeutics is a prominent one that have been using microscopy and machine learning broadly to do, so-called phenotypic screens. Um, Earlier, I mentioned you often pick a protein target,
Wait, sorry, I
and
need
you
to slow down for my ninth grade
yes,
biology. Phenotypic screen is what?
yes. So this is my apologies.
No, I'm like I know the word phenotype. It's like the expression of a gene. Is that right? I should not guess.
Yes, exactly. So I think it's like one way to think about it is maybe bottom up design versus top down design. So kind of the targeted drug discoveries may be bottom up. You say the human body is complicated. I'm going to be a reductionist. I think there's this one magic lever, and I can swip that lever on and off. I can really change everything. And that's kind of you come from the bottom, and then you hope it makes it all the way to the top. The other way, which is actually the more traditional way of finding medicine, is like some really smart doctor, like this is like the penicillin story, notes some like effect. You have no idea what the effect is caused by. You don't really understand the intricate uh, biophysics, the chemistry behind it, but you see it. Like
Mhm.
maybe there's something that just you observe in I think this famous case of penicillin in like what was it, the mold on the bread.
Mhm.
But I think for a phenotypic screen like the ones Recursion do, basically they have these cell-based assays where they grow cells in a petri dish. And essentially they test, you put a little bit of medicine in there, and then you see how the cell's state changes and use the microscope and the deep learning system on that to pick up those changes. So you can do this very rapidly.
But like, what would
And
be
then
an example change? Like the cells are a different shape? Is that?
hmm, that's a really good question. I think it often depends on. So I think it depends on the disease in question. So like a common thing, say for like cancer, is that the silly one is, can you kill the tumor
Mhm.
cells? Which the hard part there is, can you kill it without just finding bleach?
Mhm.
So that's something that's a medicine. I think for other readouts really depend on the disease. Like um, kind of, I think the general point there is like disease is complicated, so there's many proxies people use. So
Mhm.
kind of the hierarchy of proxies is, if you have a pure test tube, which is molecules, that's like the weakest. If you have cells, that's a little better. If you have a rat, that's a little better. But I think
Mhm.
the gold standard, of course, is like the human.
Right.
Um, so this is, you can think of this as like, it's better than the pure test tube, but it's absolutely not the same as like uh, but as a human, but
Mhm.
it is a useful kind of proxy.
Mhm. So okay, so then what the machine learning does is kind of find properties based on the images from the microscope.
So I think that the way I like to think about it is that the machine learning is kind of like making a better microscope. So in many ways, like, if you go back to kind of classical signal processing, like we have all these, Fourier transforms, you have high pass filters, low pass filters, what have yous. And these, traditional signal processing techniques made things like, you know, uh, microscopy even feasible in the first place. Uh, well, you had purely kind of optical microscopes back in the day. But in the last century, I think there's been a lot of signal processing attached to it. So I think of
Mhm.
deep learning in these applications as signal processing, turned up to 11. So you can pull things out of the image that you didn't. There's no obvious way to write down that function. Um, so I think right now it's more like this really fascinating scientific thing, isn't there's got to be something there. Like,
But I want to make sure I'm like picturing, like I want to have a
mm,
mental model. So like maybe that was evocative of like, did I kill the tumor cells? So is the point that like the machine learning could tell me if the tumor cells were killed without me having to actually look at it? Or is it that the machine learning like sees something deeper that like I couldn't figure out if I looked at it?
so I'll have to apologize up front because I'm not an expert at cellular biology, but I'll try to. So, for example, I think for some, I might be making this up. So, if there's real biologists that are eventually listening to this, please bear with my.
No, it's a machine
Uh,
learning audience. You can pontificate.
but I think, okay, so I think if you take some
And by the
muscle
way, I
cells.
think machine learning people will be really familiar with the idea of like just looking at results and not worrying about the process behind it. So I feel like this is really appealing to our machine learning audience.
I do have to say, I still have no idea about what happens deep in layer 37 of my column. Um, but so I think kind of coming back there. So imagine you have a muscle cell, and you can
Mhm.
often measure like some measure of like the stretchiness of the muscle cell. Like there's often ways to kind of guess at a proxy for healthiness.
Mhm.
I think the actual thing you measure depends a lot on the biology of the system. So I think for example, like one common thing is that you might, there's these things called fluorescent reporters, and you can engineer the cell so that, if you have the drug and it actually hits something in the cell that you know about, it like sets off a light. Here it's, you
Mhm.
have to know a little bit about what's happening inside the cell. You have to have a guess already. um, I think the cruder version might be like, you have this muscle cell, you're looking at, maybe there's some measure of how stretchy it is. Oftentimes, oftentimes it's just like kind of obvious to the eye. It's like that traditional, like, you know a dog when you see it, as in you see the healthy
Mhm.
cells, they have some like nice geometric shape, it looks good. Then you see like disease and they're all like shriveled up and just looks
Uh-huh.
bad. And you
Yeah.
can't quite write down that function, but you kind of know
Yeah.
when you look at it.
Yeah.
um,
Totally.
so it makes sense to nets can begin to pick this up.
Right. And I guess I've seen versions of like cancer cells and like kind of different levels and what do they call them, like biopsies,
Mhm.
where you look at the cells. This is ninth grade biology, so I should probably cut this out. But yeah, I guess I can picture what you're saying of like there's like healthy cells and
Yep.
but is the point then? I guess what my question is, like, what is the machine learning helping with? Like, is it sort
Mhm.
of like reducing the cost of looking at this stuff, or is it like pulling out other signals that are somehow like useful?
I think it's a bit of both. So I think traditionally, the traditional labor was you'd have a grad student whose painful job it is, if you're unfortunate enough to be stuck in this lab, to look at cell 1, 2, 3, 10,000. And now I think that it's often there are I think a number of readouts where it's that thing where you just look at it and you kind of know there's a difference. So I think you can train yourself to like read these things. Whereas
Mm-hmm.
I think this is again I think a lot like the Pinterest example you brought up where you're training the model to um, basically pick out something and you do it at bigger scale. So maybe before I could only test 10,000 because the grad student union would revolt at that point. Um, but now maybe I can test a billion, or
Mm-hmm.
I'm limited more by my supplies. I think the second question you asked is actually the more exciting one, as in, is it possible we can pick out something we didn't know?
Mm-hmm.
So I think there's glimmers that this is yes. I know there's a few papers that are doing things like you can identify where the organelles are. You can begin to do some more complex readout, but I think there is sort of almost a chicken and egg problem here, as in like when you're discovering something, you kind of have to like it's like unsupervised learning, right? If you know the thing you're looking for, then you can like slot it into buckets pretty easily.
Mm-hmm.
But then if it's like you want to go deeper and find something you don't
Mm-hmm.
know, so I think yes, I think there are likely places the quantum nets, act as like amplified microscopes and like pick up biology that we don't know. But if I knew that, I would have gone off and written a Nature paper about it already. So I think that's, or I'm sure there's a couple that have already come out in this vein.
Okay, well, so I have to ask you. I mean, one of the Nature papers that blew my mind, and I think a lot of people, was the one, the dermatologist one, where they fine tuned an ImageNet classifier on cancer. So that was not like under microscope, right? That was just literally just like photos,
Yep.
and that seemed like so amazing. I mean, should I be as enamored with that as I felt, or is there some gotchas where it's not actually should we actually be like using doctors for these diagnoses still, or it sort of seemed like from the paper that it was more accurate than the doctor's diagnoses, wasn't it?
I think that entire field for sure. I think is like radiology or I think usually it's yeah like pathology or you like dermatology. You look at some picture and then you kind of diagnose it. I think that absolutely is a place convents will just make a big difference. And I do think that these models do kind of achieve a striking advance over what you could do previously. Um, so my understanding though is that the challenge there is that like sometimes these models pick up things that are kind of silly. I remember there's this like very excellent blog post written um, where it kind of discussed failure modes where it turned out there's some like scans from different trauma centers and the model is doing an amazing job, 99% accuracy. And anytime you see that 99% accuracy, something is up. It turned out there's like some label at the bottom or something that printed the trauma center, so there's like light trauma, heavy trauma, and guess what that model learned to do right there.
Uh,
um, so I think is it kind of comes down to what is the model learning? Is it
mhm,
is it a fluke? Is it kind of an actual thing? Radiologists are kind of tried and tested. Like, do you really want to fire like your world class radiologists? Like, so I think there's a natural caution there. I think in part because we don't really understand what happens deep in like layer 37 of the ResNet.
mhm,
Um, so I think the FDA and like some companies are moving forward. I do think in potentially in places where there aren't enough doctors, this could be kind of a potentially revolutionary advance where you could get world class scanning centers available, clinics throughout the world, and not just places where you have excellent hospitals already. But I think I think it'll take some time. I remember a number of years ago, I think maybe in the 80s again, there's a whole wave of hype around expert systems for medicine. and how they could diagnose patients. And I think this might have been that same blog, a retrospective study that found that in many cases, hospitals that deployed expert systems actually had a fall in patient kind of well-being afterwards, because there were these complex interactions that no one thought of in the first study, and then you find a number of years later that there's just unexpected side effects. So, yeah, I think I am, long-winded answer there. I think it is something to be interested in, excited about. I think it will also take time to really vet and really kind of like make sure that this is something that improves patient well-being.
Although I guess, I mean, do you know like what happened with the that melanoma model? Because it does seem like, doctors are also not perfect, and I also cannot inspect my doctor's brain to know
Yep.
really know their decision making process. So I wonder, is it unsafe to not change, or was there some real flaw, or some simplification that
So
wasn't obvious?
I don't think there's a flaw in the paper. My guess is that this isn't my field, so I'm kind of projecting a little bit out there. I know that, um, the entire, deploying something in the clinic and the healthcare side is actually, I think, quite more complicated even than the new biotech side. I think you have to work with insurers, you have to work with payers, you have to work with hospitals and doctors. So I think, the American healthcare system has many known challenges. I my sense is that this has just been very hard to actually get out there. So I think, in pharma and biotech, I think the advantage is like if you get something and it works, there is actually a very well known to like path to get it to people. I think for advances like this dermatology thing, there's actually a fuzzier, more ill-defined path to get it out there in the wild. Um, so I think it doesn't have. I think there are some real scientific questions around: is this actually robust that are still unanswered? But I think there's
Mhm.
also harder business questions about: does this make sense? Is it a viable business? And I'm sure there's like a dozen startups who are working on this right now, but I just don't know as much about it.
It's funny. My wife runs a healthcare
Ah,
startup, and she tells me that it's the only industry where you could literally save money and save lives simultaneously and not have a viable business.
I've had a few friends who left healthcare and have formed, ostensibly boring but very successful startups and are much happier with their lives. So I sympathize just a little bit. But you probably know way more about this than I do. Like it's a little bit outside kind of my expertise. But yeah, it
No,
seems
sorry
really
to take you
challenging.
outside of expertise, but this is what I was hoping with the podcast. I could corner guys like you and ask all my dumb questions. So I really appreciate it. And I think we should kind of wrap up, because I think this might be just getting long for the format. But we always end with two questions that I'm kind of curious, actually. I always say this, but really I'm curious how you're going to answer these. So, what is one really underrated aspect of machine learning that you think people should pay more attention to?
Hmm.
What comes to
That's a,
mind?
that's a really good question. I think that machine learning as amplified signal processing, I think, is a view that is not as commonly celebrated. But I think there's these really exciting things going on where, machine learning is like finding its way into instruments, like into sequencers, into microscopes. Um, it's a type of Internet of Things, you could say, but like not the consumer version. But, I think traditionally new scientific instruments always are the, uh, predecessor to like fundamental new scientific discovery. So I think that when we find deep learning is making our instruments better, more capable, then I think that we're actually setting ourselves up to discover new fundamental science. So that's something I'm very excited by, but it's kind of a longer, you know, we might have the instrument, but we still need, the Einstein or something to come in and, work that and really get us that magical new understanding about the world. But I'm excited by that.
That's a totally cool answer. But I guess they might give so many readings that it's like hard to even interpret. But I guess a good algorithm would give you a few high values. What do you call them? Say, processed outputs.
I think yeah, it's definitely. I think for now it's still going to be quite a while before I think we see. Um, I think we talk a lot about HAI, and I know there's many ways in which you could get a general intelligence, but I think the process of induction of interpolating things about reality from very few you know hunches. This is probably made up, the Newton and the apple tree. Yeah, like if it probably didn't happen that way. We know it's the just so story, but you could imagine some machine learning model like, seeing that, and can you somehow interpolate from that out to a universal law of gravitation? But yeah, that I think would be amazing. It just seems far beyond our current science.
It's funny though. I feel like with all these medical applications, I guess the reason I naively find them exciting is that, like if you're trying to compete with the human for navigation and driving, it's like, boy, our brains are designed for that, right? Like clearly, like huge part of our brain is just to
Yep.
navigate the world and not crash into stuff. But it doesn't seem like our brains are designed for like interpreting molecules that we can't see and like what effects they might have. I mean, I'm still trying to visualize it in my head, and it's I can't even do it. So it sort of seems like maybe the bar is lower for like a useful algorithm.
That's I think that's a really interesting kind of point there. I do think understanding quantum mechanics is kind of at least it doesn't fit in my head. It's kind of there's lots of complicated things going on in that hidden world there. Like maybe part of the challenge is that it's hard to validate like a discovery. Like many times the model says something, but after you spend a while, like 9 times out of 10, you're like, all right, what bullshit did the system pick up this time? Um, and I think the challenge there is like um, maybe we have to make the model, like you said, we have to make the models robust enough that there's actually high quality signals coming out. So we're like, oh, that's a clue, and not, oh, I don't know what hiccup happened in step 2000 of like gradient descent. So I think that's maybe the challenge where we just haven't. I think this was beginning to change, but it feels like still discovery, like invention, is the province of the human and not the machine. But maybe
Yeah,
that's like the antiquated line, and 10 years from now you'll have AI discovered everything, and I'll be like, well, that aged poorly there.
we'd be an interesting world if that comes to pass. Sorry. So final question is, so right now in 2020, and I guess it's already June. What do you think is currently the biggest challenge of making machine learning models work in the real world? Like in your
That's a
experience, like what
great
are the
question.
challenges that you've run into? Like what have been the surprising hurdles?
I think things more specific to me are often small data.
Mhm.
Like again, you have 30 data points. And oftentimes it's a very well-meaning scientist who kind of comes and says, "What can you do for us with 30 data points?" And then
Yep.
oftentimes I'm like, I wish I had a better answer. Sometimes you just try 7 things, like you try the transfer learning, you try like the multitask learning, the meta learning, and all the
Mhm.
learnings fail. And then at the end, the random forest is like, yeah, it's not great, but it does something. So I
Mhm.
think for things I'm excited by, I think like robust transfer learning that actually works on small data, which I
Mhm.
think is has occurred in NLP,
Mhm.
but I think has not occurred in molecules. I think that would be an amazing advance for this
It's
field.
so interesting it hasn't occurred because I feel like it's also totally happened in vision, like for
Yeah.
sure, right? And NLP now definitely it's a fact.
Yep.
It's,
Yep.
it's
Yep.
so interesting it doesn't work for molecules.
It might just be data. Like I think if someone just found, a gigantic, trove of molecular measurements that was high quality and you had a billion of them.
Yeah, or collected it. Nobody's gonna find that, right?
I think this is the sort of thing that I think a governmental effort could do like amazing work at.
Yeah.
And to be fair, I think like governmental agencies have actually put out most of the open source data out there, so they are actually working hard at this. But yeah, there's maybe the sort of thing that like if you get a $10 million grant or something, I think you could make a serious dent at putting together a high quality open data set for this. But it is just more expensive than ImageNet, and it will take more resources just because you need to do the actual experiments.
Mhm. Great answer. I love it. Um, well, thank you so much. Is there like some place we should tell people to contact you, or is there anything you want to promote? Deep Chem, everyone should try it.
I think absolutely. I think part of like the goal behind DeepChem really is to make open source more feasible for drug discovery. So I think we could definitely use more users. In particular, if you're an engineer that knows how to handle build processes, well, please get in touch. I'm trying to figure out the Windows and etc. builds, and it's just such a pain.
Oh man,
Too much of
you're
a
telling me.
scientist. But yeah, we could absolutely use more help. So if you're interested in open science, yeah, please do get involved.
I love it. Thanks, Bharat.
My
Awesome.
pleasure. Thank you for inviting me.